Hi everybody, welcome to another Chats with Chaudhry. I hope you're all safe and well. Today I'm delighted to be joined by Andrew Basset, who is Head of Research of Cellular Operations at the Wellcome Sanger Institute, and he's also co-chair of the upcoming ELRIG CRISPR in Drug Discovery event. And that's what we're going to be talking about today, which is next week's event. So Andrew, lovely to meet you. How are you?
Yeah, lovely to meet you too, Rizwan. I'm very well, thanks. How are you?
I'm very, very well, although this week is a bit up and down in terms of weather. 1 minute it's raining, next minute it's bright sunshine. You never know what to wear when you walk out the door, do you?
That's true. Yeah.
Well, I'm very well apart from that. Look, we're obviously going to talk about next week's event. Before we do that, could you give people who are not familiar with ELRIG a quick overview of the organisation?
Okay, so I mean, ELRIG is I think the official title is European Laboratory Research and Innovation Group, which is a bit of a
Thank you for
mouthful.
doing that.
but it's a not-for-profit organisation that aims to bring together the drug discovery and the life sciences communities at a variety of different events. So they hold a program of about 6 or 7 events in a year, which are a mixture of mainly conferences, but also networking and webinar style events. And they're incredibly, they're sort of world-class events. I think the conferences are anyway. And I think sort of one of the really nice things that I've found about it is that it brings together the academic communities and the industry communities and gets them talking with each other. I think one of the big things also is that it's all free. So all of these things are meant to be readily accessible to everybody, and you can just turn up and come along. So they make all the events free to join. Yeah, I think they've been really good in my experience, actually, and I've really enjoyed a lot of the ones that I've been to. So.
Fantastic. And your role as co-chair on the committee for the CRISPR event. So what does that involve?
So basically, we sit down and we work out who we want to invite,
Right.
what sort of subjects we want to include on the program, what we think is exciting in the CRISPR world, and we try and get those people to come along and take part. So it's obviously been a bit different this year. It's kind of because this is a virtual event in this case, but that's actually meant that we can reach out a little bit more broadly. So we can pick people who wouldn't necessarily want to make the sort of trip from the west coast of the States or from halfway around the world, but actually they're quite willing to do a webinar. So I think we've got a really nice selection of speakers. Actually, I think it's great.
Fantastic. We'll talk about those shortly. So let's start though with a very basic question. I'm not a scientist, as you can tell, obviously, by some of the questions I'm going to ask you. But for those who are not familiar, what actually is CRISPR and why is it so important for drug discovery?
Yeah, so CRISPR is it's one of these sort of horrendous acronyms. So it actually stands for Clustered Regularly Interspaced Short Palindromic Repeats, which tells you nothing at all about what it does. So it's actually a bacterial defense system against viruses. That's how it was discovered.
Right.
But that actually turns out to be an enormously useful tool, because the way that works is it captures a bit of viral sequence and it uses that as a guide to target mutations or DNA breaks in the virus. And what the sort of realization was, sort of, I think it was about 2013 now, was that you could actually transfer that into almost any other system, including human cells or other cells. So basically what you have is a way of being able to specifically make DNA cuts in the genome, wherever you want in the genome, just by changing a short 20 nucleotide sequence of what's called guide RNA. And that just base pairs with the bit of DNA that you're interested in. So it means that you have a sort of a programmable tool for making breaks in the genome. So that's kind of pervasive across the whole field of drug discovery now, and life sciences in general, I think, because it's meant that firstly you're able to make genetic changes in organisms that you wouldn't have been able to beforehand, like human cells is a good example from the point of view of this. That sort of has meant that it's been used sort of in every aspect of that kind of drug discovery process. So from everything from the target identification side of things, target validation, so making better models of disease, through to even the therapeutics now. And I think those kind of 3 aspects are what we're sort of trying to do in this conference. We kind of run through from one end to the other, if you like.
Right.
Yeah.
Brilliant.
So
And sorry, Karen, please.
no,
Sir.
okay.
So what I was going to say, so what are the main challenges though in terms of CRISPR as a technique then? Because there must be some challenges depending on what areas you're using it in.
Yeah, definitely. I mean, I think you're absolutely right. It depends very much on the area that you're looking at. So, you know, from the sort of target identification point of view, we're quite good now at doing even genome-wide screens, and there's a very nice talk sort of in the next week about sort of how this has been applied to a lot of different cancer cell lines. So we can knock out every gene in the genome and understand what function that has. So I think the kind of the ways that's progressing now in that area is kind of making it more complex types of mutations, so not just knockouts, more complex mutations in better model systems, so things that better model disease, the more complex sort of 3D cultures and other sort of cell systems. And then with more complex readouts, so sort of, you know, we're starting to get into sort of things like single cell sequencing as a readout of these screens, or more complex, not just life or death readouts, but actually more subtle sort of phenotypic readouts for these. So I think that's kind of the way things are developing in that area, I think. Whereas if you're looking at something like therapeutics, it's a completely different sort of set of problems, really. You know, they're the issues are more in terms of how do you deliver it, how do you make sure it's not going to do something nasty, so it doesn't have an off-target effect somewhere else in the genome that you don't want it to, and it causes a mutation that gives you cancer or whatever it is. So I think there's kind of a need for sort of it to be highly specific, highly targeted, without any kind of side effects. So there's a very different sort of set of problems that they are to sort of solve in that area as well.
Right. And one topical question, I mean, obviously everyone's been talking about vaccines and so on. So has CRISPR been used in terms of the development of the vaccines, and how?
Yeah, so I think mainly sort of its uses in the sort of screening for the proteins and the genes involved in viral infections. So so people have certainly used it to identify those genes which are key for viral infection and for viral sort of function. And I think that's been enormously informative in terms of understanding the pathways and the receptors that sort of that recognize the virus, and sort of the different pathways that are being activated downstream. So yeah, I think it has been hugely useful.
Brilliant. Thank you. Now, so let's talk about the conference next week. What are some of the highlights then from your perspective, and why should people, even, I mean, free or not, and it's great that it is free, but why should people spend their time coming to the event next week?
Yeah, so I think I'm really pleased with the set of speakers we have. I think it's an excellent group of speakers. I think, you know, and these it's kind of split into three areas. So we have the functional genomics bit,
Right.
and we have some really nice sort of, and the keynotes are very good, but I think a lot of the other talks sort of kind of play to some of the other sort of aspects of things. So for instance, we've got sort of a very big scaled sort of genome-wide screening project from DepMap. There's kind of some of these ideas of more complex readouts. So we've got a couple of people talking about single cell readouts, more complex transcriptomic readouts for screening, and you know, even getting into sort of more complex sort of single nucleotide changes of mutations. So I think that's all really exciting. We've got a section on kind of modeling of disease and kind of, you know, how do we do that? That's mainly sort of iPSC derived models, 3D models. We have Madeline Lancaster talking about sort of mini brains and sort of how we can use those for disease modeling. And then the therapeutic section, I think, is also enormously exciting. I mean, it's amazing to watch this happen as sort of these things are being translated from work in the lab to actually treating people with genetic disease, and not just sort of treating them, but actually curing the genetic cause of that disease. And again, we have some really sort of exciting sort of people who are, you know, really quite on the cutting edge of sort of being able to do that, and actively physically doing that in humans now. So I think it's an exciting meeting. I'm certainly excited by it. So.
Fantastic. Well, thank you for giving me that summary and for giving me that introduction to CRISPR as well. So I really found that useful. So if you are interested in CRISPR, in any aspects of CRISPR, then you should make sure that you sign up for the ELRIG event next week. It's running from the 23rd to 24th of March. So it's 2 days. And as Andrew said, it's completely free. So all you have to give up is your time, but it's time well spent because there's a very active program. There's lots of people attending. I mean, I know usually the ELRIG events get quite a few hundred people coming to them over the 2 days. So it's definitely worthwhile going, not just in terms of seeing the content, but also networking and connecting with other people who've got similar interests as yourself. So the links will be above the video, so you can actually find out more about the event and register. And if you've got any questions that Andrew's discussed today about the areas Andrew's discussed, then feel free to leave comments below the video, and I'm sure Andrew will be happy to answer them for you as well. So Andrew, I appreciate you taking the time to do this. I know it was all very last minute for you to do this, so I really appreciate you taking the time out and answering those questions for me. And I wish you success in the build up in the next few days, because it's always stressful as you lead up to an event to make sure all the website and everything is working and functionally properly, and everybody else is going to be there on the day. So
we have a very good team to help us with that. So
yeah, fantastic. Well, good luck with the build up and have a fantastic event next week. And I look forward to attending myself and seeing more in terms of the sessions and networking with other people as well. So thank you very much for your time.
Thank you. Thank you very much as well.
And thank you all for watching. As I said, if you've got any questions, please leave them for Andrew below the video, and make sure you sign up. The link will be above the video. Thanks a lot. As always, stay well and stay safe. Bye-bye.